Good morning. Let's talk on digestion and absorption of protein. We know proteins are very essential for the sustenance of life. Proteins are the large globular molecules and their chemical breakdown requires time and mixing. The protein digestion in the stomach takes a longer time than carbohydrate digestion but a shorter time than the fat digestion. Today's learning objectives are to describe the digestion, absorption, and transportation of dietary proteins with associated disorders. Proteolytic enzymes break down the dietary protein into their constituent amino acid. These enzymes are produced by stomach, pancreas, and small intestine. There is no digestion of protein in the mouth. Digestion in a stomach. When the protein enters the stomach, the gastrin hormone stimulates the release of hydrochloric acid, pepsinogen, and renin in infant in the stomach. The hydrochloric acid denatures the protein, making their internal peptide bond more accessible to subsequent hydrolysis by proteases. Provides an acid environment for the action of pepsid. These are the two important functions of hydrochloric acids. Pepsin is secreted as pepsinogen, which is in the inactive form. The pepsin cleaves those peptide bonds of protein involving the aromatic amino acid and acidic amino acid. These few aromatic amino acid are phenylalanine, tyrosine, and tryptophan, and acidic amino acid are aspartic acid and glutamic acid. Pepsin cleaves the long peptide polypeptide chains into the mixture of a smaller peptide and some free amino acids. Renin is absent in the adults, only it is present in the infants and also called the chymosin or renin. Action of renin is to clot the milk. It converts by this reaction the casein of the milk. It's under the influence of renin hormone, renin enzyme, it converts up into the paracasein, and paracasein along with the calcium ions, it converts up into insoluble curd-like curd, that is the calcium paracasinate. So you can see it here, the food we have taken, it first of all reaches to the stomach and there is no digestion starts in the mouth. For the protein, digestion starts in the stomach. So once the food reaches, the hydrochloric acid starts secreting out along with pepsin in the form of pepsinogen. And if it is the infant, the renin hormone is also renin enzyme is also being secreted. HCl activates the pepsin and pepsinogen converts up into the pepsin, which is now the active in the active form. It converts, partially digests the proteins into smaller peptides and protein, and these, after some stay in the stomach, they are finally shifted to the small intestine, and in the small intestine the pancreatic secretion starts pouring out, and under the influence of bicarbonate, the whole pH of the environment changes to the basic, right from acidic environment of the acid. So the pancreatic juice contain the following enzyme: trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidases A and B. So you can see it here. The pepsinogen is converting into the pepsin under the influence of H plus ion. This is the stomach reaction of the stomach, which I have already explained. Then these are the enzymes secreted by the pancreas, uh, and these are trypsinogen, chymotrypsinogen, proelastase, and procarboxypeptidase. So trypsinogen under the influence of enzyme enteropeptidase, the enzyme which is secreted from the intestinal wall, and it converts trypsinogen into the trypsin. Once the trypsin is being activated, all three enzymes are being activated by the trypsin. So trypsin has a vast role here. So trypsin is converting the chymotrypsinogen into the chymotrypsin, proelastase into the elastase, procarboxypeptidase into the carboxypeptidases. All the dipeptases complete the digestion of dipeptides to free amino acid, which are also present in the intestine, secreted by the intestinal fresh water. So we can see the site of action of different enzymes. Uh, The aminopeptidases secreted from the intestinal lumen. Their site of action is at the termini of the end termini of the peptidic chain. This is the whole peptidic chain. Small chain is there. So this is the end terminal end, and this is the carboxy terminal end. So at the amino terminal end, these aminopeptidases are breaking the peptide bond. Then pepsin, the seat of action of pepsin is at those peptide bond which are being contributed by phenylalanine, tyrosine, glutamine, and asparagine. Then trypsin is the seat of action, it's breaking those peptide bonds which are contributed by the arginine and lysine. Similarly, chymotrypsin is breaking those peptide bonds which are being found by phenylalanine, tyrosine, tryptophan, and leucine. And elastase is breaking those peptide bond which is being contributed by adenine, glycine, and serine. Then carboxypeptidase is again of two types, carboxypeptidase A and carboxypeptidase B. Carboxypeptidase A is breaking those bonds which are being contributed by those amino acid which are having the hydrophobic bulk, while the carboxypeptidase B is attacking on those peptide bond which are contributed by arginine and lysine. So this is the seat of action of various enzymes at various sites of peptide chain. So after being absorbed, the dipeptides are, once the dipeptides are left and smaller di and tripeptides, they are being finally broken up by dipeptidases enzyme, which are being secreted by the intestinal wall, and they completely digest all the proteins into all the dipeptides into a smaller individual amino acid. Now let's have a look onto the absorption. So for absorption at the intestinal wall, there are in the intestinal epithelium cell, there are number of provisions available in the form of various transporter. So this is the intestinal lumen. You can see it here. This is the lumen side, and this is the intestinal epithelial cell, and this is the serosal side. And we can see the pink color, the portal vein is there. So we can show you here. This is the co-transportation of amino acid is happening along with the sodium. So this is a kind of secondary active transport. Sodium is facilitating the uh, transportation of amino acid, but it requires ATP, the energy, and the energy is indirectly contributed by the sodium potassium pump. You can see sodium potassium pump. Sodium which is being entering here in the epithelium cell is now uh, removed out of into the lumen with the help of sodium potassium pump. So three sodiums goes out, and in return the two potassium are taking their entry in the epithelial cell, and in this course of transportation, one ATP is broken up into the ADP. The energy which is being released is utilized for this pump operation as well as for this secondary active transportation. So amino acids, once being entered into the epithelium via facilitated transportation method, and this is the facilitating proteins available, and that is transporting the amino acid, individual amino acid into the portal vein, into pouring into the blood. So this is how absorption is happening. Another very interesting cycle is there, which was being explored by Meister, who proposed that glutathione participates in the absorption of amino acid in intestine, kidneys, and brain. And the cycle is called as gamma glutamyl cysteine cycle or the Meister cycle. Glutathione is, I've already explained why in my various lectures, like it is a tripeptide and it is in so much of abundance present into the cell. It is actually the gamma glutamyl cysteine glycine. So here the isopeptide bond is there, gamma peptide bond is there between the glutamyl and cysteine. So this is the speciality of this tripeptide, and it is participating in the absorption of amino acid. So this is the ischemic presentation. This is the amino acid at the extracellular side, or we can say in the lumen of intestine. So what is happening here? The amino acid winds up with glutathione in the presence of enzyme gamma glutamyl transpeptidase. So in this way, 1 ATP is utilized here, and this glutathione is broken up into cysteine glycine portion. One portion is cysteine glycine, and the glutamyl gamma glutamyl portion, it catches the amino acid and it makes the adduct gamma glutamyl amino acid. So gamma glutamyl amino acid is entering the epithelium cell, intestinal epithelium cell, and gamma glutamyl amino acid under the influence of another enzyme, that is the gamma glutamyl cyclotransferase, it breaks up the this adduct into the to free the amino acid, and the glutamyl portion is converted up into the 5-oxoproline. At the same time, the cysteine glycine is also entering the epithelial cell, and within the cell, this cysteine glycine is broken up into the glycine and cysteine with the help of this enzyme dipeptidase. So oxoproline, which is being released here, again being converted up into the L-glutamate by the enzyme 5-oxoprolinase, and L-glutamate combines with cysteine, leading to again generation of gamma glutamyl cysteine, and this gamma glutamyl cysteine again along with the glycine, and the enzyme is glutathione synthetase, it regenerates the glutathione. So this is how the glutathione is recycled. It goes out into the lumen of intestine, again catches another slot of amino acid, and this process goes and goes on. So number of kinds of amino acids are being transported, absorbed and transported through this method, but there are few drawbacks. Like this pathway is only restricted to intestine, kidneys, and brain. It is not available at all the sides of the body. Secondarily, it requires a huge amount of ATP to be invested. Exhaustion of ATPs happen. 3 ATPs are lost here in this process. Another thing is that few amino acids like hydroxyproline and proline cannot be transported, cannot be absorbed by the Alton-Meister cycle. Now, absorption of intact proteins. So, a small intestinal cell of fetal and newborn infants are able to absorb intact protein. For example, the immunoglobulins in infants can be absorbed. IgA from the colostrum of milk are absorbed intact without the loss of biological activity, so that they provide the passive immunity to the infant from the intestinal cell. The intact proteins are not absorbed by the adult intestine, so that is not that characteristic is not available among the adults. So here there is a table showing the amino acid transporters and their associated disorders. So transport system for small and large neutral amino acids. So amino acids are alanine, serine, and threonine, isoleucine, leucine, valine, tyrosine, tryptophan, and phenylalanine. If there is a defect in this transporter, lead to the disease Hartnup disease. Secondarily, the basic amino acid and cysteine transporters, if there is a defect, so there is a lack of transportation of these arginine, lysine, and cysteine, and leading to the cystinuria. A lot of cysteine is going through urine instead of absorbing at proper site. So acidic amino acid transporters, they are meant for glutamic acid and aspartic acid. Similarly, the amino acid and glycine transporters, they are transporting protein, hydroxyproline, and glycine. So in absence or deficiency, or there is some disorder associated with this transporter, will lead to the glycinuria.